Welcome to the talk on the early development in nematodes. Caenorhabditis elegans or C. elegans is a free-living worm which is a hermaphrodite. That basically means it has both male and female sex organs. It belongs to the phylum Nematoda and is widely used as a model to study many different biological processes which include development. The adult C. elegans has 959 somatic cells. It is able to undergo embryogenesis within 16 hours, and hence its developmental processes are relatively quick. It is also easy to genetically manipulate, and all these factors enable C. elegans to be used as a model for experimental processes. So let's look at what actually happens during the fertilization process in C. elegans. So Caenorhabditis elegans, as I told you before, is a hermaphrodite, which means it produces both sperm and eggs, and it can thus undergo self-fertilization. So during the process of fertilization, it's pretty interesting because what happens is the eggs actually roll through a chamber called as the spermatheca. So if we look at the figure and the blue box, that is where the spermatheca is. The spermatheca is where the sperm are present. Now we can see there in the C. elegans body, there are actually two spermatheca. And when we look at right next to the spermatheca, we actually will have the oocytes that are in different stages of development. And as they move closer and closer to the spermatheca, the more and more they are developed. The eggs will actually roll through the spermatheca, and after they roll to the spermatheca, fertilization occurs and you'll start having development of the embryo within C. elegans. So after the embryo undergoes early cell divisions, it will leave the organism through an opening that's called as the vulva, which is showed right here. Thus, in the case of fertilization of C. elegans, the eggs will actually roll through the spermatheca to form the embryo, which undergoes early cell divisions and then leaves the organism through the vulva. The zygote of C. elegans undergoes rotational holoblastic cleavage. This is different than what we observed in snails, which underwent spiral holoblastic cleavage. So let us see what happens during rotational holoblastic cleavage. Now during the first cleavage step, the zygote undergoes asymmetric cell division to form a daughter cell that is called as the anterior founder cell or AB cell, which is larger than the second daughter cell, which is called as the posterior stem cell or P1 cell. Now to understand rotational holoblastic cleavage, we're going to see what happens during the second cleavage. During the second cleavage step, the two cells, which is the AB cell and the P1 cell, they will undergo cell division at axes that are perpendicular to each other. And so to visually see this, here we have a picture where we have both cells. The AB cell is present on the left side and the P1 cell is present on the right side. Both cells are undergoing cell division, but look at the location of that mitotic spindle. So the centrosomes are shown as these very bright green structures in both the cells, or those yellow dots. Now when we look at the plane of cell division for the AB cell, I have shown that plane in the form of a purple line. If we try to find the similar plane of cell division that is occurring in the P1 cell, as you can see, that purple line is perpendicular to the purple line that's present for the AB cell. Thus, since the axes are perpendicular to each other, there is a rotation in the axis of mitosis, and that is why it is called rotational cleavage. The product of the second cleavage is going to be four daughter cells called ABA, ABP, P2, and EMS. ABA and ABP comes from the AB cell, and P2 and EMS come from the P1 cell. Now let's look at how the dorsal ventral axis is formed in C. elegans. Now when the AB cell undergoes cell division, the daughter cells do not have enough space to remain side by side. So when we look at the figure in the blue box, this is where the AB cell is undergoing cell division. And technically the daughter cells will basically be one on top of another. However, there's not enough space to have both of them remain exactly one on top of another or side by side. And so the cells will slide in such a way that the ABP cell lies to the posterior or to the back of the ABA cell, and that is why they got their names as ABA and ABP. ABA is the anterior daughter cell, whereas ABP is the posterior daughter cell, and that is what we can see here in the figure in the blue box. Now let's pay attention to the P1 cell. When the P1 cell undergoes cell division, it will form the daughter cells P2 and EMS. Now when we look at the location of P2 and EMS, EMS turns out to be right below the APB cell. This arrangement of the 4 cells in the embryo helps establish the dorsal ventral axis. The ABP cell forms the future dorsal side, while the EMS cell forms the future ventral side of the organism. Now let us see how anterior-posterior axis is formed in C. elegans. Now the anterior and the posterior part of the embryo is actually determined by the point of sperm entry. The place that the sperm enters the egg becomes the posterior end of the embryo. Now the reason this happens is because the sperm microtubules aid in the differential localization of a class of proteins called as the PAR proteins. So if we look at the figure, the part in the blue box, let us say that is where the sperm entered the egg. And that is where we can now find the sperm centrosome as well as the sperm's pronucleus. That part of the egg now becomes the posterior part of the zygote, and the diametrically opposite part where the egg pronucleus is becomes the anterior part. Now the reason this axis gets formed the way it is after the sperm has entered and the centrosome is present, what happens is the microtubules start emanating from the sperm centrosome. These microtubules start attracting certain groups of the PAR proteins, and other groups of the PAR proteins move towards the egg portion, where the egg pronucleus is. Thus, when cell division happens, we will see that there is a distribution of the PAR proteins in such a way that the AB cell will have the PAR proteins PAR6 and PAR3, whereas the P1 cell will have the PAR proteins PAR1 and PAR2. Thus, we can see the differential expression of PAR proteins in the AB cell and the P1 cell, and this is what helps form the anterior-posterior axis in the case of C. elegans. Now let's look at the processes of specification. Now in a 2-cell embryo, if we remove the P1 cell and allow it to develop independently, it will form all the cells that it would normally make in the intact embryo, which is the posterior half of the embryo. Since it already has the ability to know what it is going to form, the P1 cell has undergone autonomous specification. Now, what if we took out the AB cell? So if we remove the AB cell and allow it to develop independently, it is unable to make all the cells that it would normally make. Thus, it does not undergo autonomous specification, but undergoes conditional specification because it needs to get cues from neighboring cells to determine the cell fates. So let's look at autonomous specification of the P1 cells. Now, the P1 lineages will have cytoplasmic factors inside them that aid in self-aid determination autonomously. Some of the transcription factors that help in autonomous determination are SKN1, PAL1, and PI1. Now, all three play important roles in determining cell fates of the P1-derived cells. And let's look at the example of one such transcription factor, which is SKN1. This is a maternally expressed transcription factor that controls the posterior pharynx formation. So when we look at the wild-type embryo where we have SKN1 present, and we look for where we can find the pharynx muscles, the part that's showing in these bright white structures are basically the pharynx muscle region. Now let us look at what happens during whether these muscles develop when we have an SKN1 mutant. So in the case of the SKN1 mutant, we can see there is no pharynx muscle development present. And thus SKN1 is important in the formation of the posterior pharynx and its muscles. So this way, transcription factors present in the P1 cell aid in the autonomous specification of P1. Now let us look at an example of conditional specification in C. elegans. Now the P1 cell will undergo cell division to form the P2 and the EMS cell. The P2 cell that is formed aids in the cell-fate determination of the ABP cell. Just to remind you, ABP cell is the cell that arose from cell division of the AB cell. Now when we look at in the figure the location of the P2 and the ABP cell, they're actually right next to one another, and hence the P2 cell can interact with the ABP cell. However, it's too far away from the ABA cell and hence it does not interact with the ABA cell. If the P2 cell is removed from the embryo or is killed in the embryo, the ABP cell cannot develop into all its cell types, which are the neurons and the hypodermal cells. The reason being that P2 is not giving the cues that is needed for the development of ABP. Some proteins that play a role in the development or in the interactions between P2 and ABP are the Notch proteins or proteins involved in Notch signaling. So there is a protein called APX1, which is actually a delta-like protein that is present in the P2 cells. This APX protein will interact with the Notch protein called GLP1 that is present in the ABP cell, and this helps determine the cell fate of the ABP cell. Now it should be noted that the name of proteins that we know in human systems like Notch and Delta, they have different names in different organisms. So in the case of C. elegans, GLP1 is a name that is used to a protein that is similar to the mammalian Notch, while APX1 is a protein that's similar to the delta-like protein that is present in mammals. After cleavage occurs, gastrulation begins in C. elegans, and it happens in the 26-cell embryo. Now, in this embryo that is made up of 26 cells, the cells will move and position themselves correctly so that organogenesis can begin. An example of such a movement that we're going to see is that of the E cells. So the E cells are shown in the figure below in the form of two cells that are shown with a yellow colored asterisk. They will actually move inwards, which is basically ingression, and eventually they form the gut. So to form the gut, they have to be inside the embryo, and hence they move from the surface area that we can see at the 0-minute panel, and they start moving, we can see at the 10-minute panel that they actually move more inwards. And as we go from the 15 to the 20-minute panel, we can start seeing that they're more and more in, and there are cells that have now come and surrounded them on the downward side. Another event that can happen is the migration of the P4 cell to a place that is beneath the gut primordium. So if we look at the figure that is shown here, we can see that the E cells, which is the cells that I had just talked about, are actually now present towards the middle of the embryo. And when we look at the P4 cell, the P4 cells also moves into such a way that they are right beneath that gut primordium that's formed by the E cell. We can also see the movement of other cells. We don't have to remember any of these movements, but you should know the concept that during gastrulation, cell movements will occur so that cells are oriented in correct position for organogenesis to happen. Following gastrulation, organogenesis will occur and then the organism hatches from the eggs. In the case of C. elegans, the organism will go through different larval stages and ultimately develop into an adult, which will be able to form the male and female gametes and thus is able to reproduce. With this, we come to the end of our talk where we learned about fertilization in C. elegans, which is a nematode worm, followed by cleavage and axis formation. We also learned about autonomous specification and conditional specification in C. elegans, as well as gastrulation in C. elegans.